In this video, we're going to talk about DNA replication. And first, let's take a look at a strand of DNA and see what its makeup is. Alright, so our DNA strands, they're going to be a double helix. So we're going to have two complementary strands, and they're kind of wound around each other. And these two strands, they are complements of each other. So they are not identical. These two strands are going to complement each other, and we're going to see what that means in just a second. Alright, so we have our sugar phosphate backbone right here. We're going to have two of them, and they're going to be bound by DNA bases. These DNA bases are going to be connecting our sugar phosphate backbones. And there are going to be four types of DNA bases, and they are adenine, thymine, cytosine, and guanine. And they are always going to bind in a certain way. So adenine and thymine are always going to bind together. A and T are always going to bind together. Cytosine and guanine are always going to bind together. C and G are always going to bind to each other. And I always remember that my aunt likes to go camping. Aunt starts with A, ends with a T. A and T, adenine and thymine, are always going to bond to each other. And camping starts with a C, ends with a G. Cytosine and guanine are always going to bond to each other. And these are our base pairs. So our base pairs, this A and T, C and G, they're going to be connecting these sugar phosphate backbones. So now let's take a look at the actual DNA replication. So we're going to start with initiation. That's when our DNA replication is going to begin. And an enzyme called helicase is going to come in, and it's going to unzip, it's going to unwind these two strands of DNA. And it's going to separate these base pairs. After that happens, or while it's happening, we're going to have elongation. And an enzyme called DNA polymerase is going to bring these free nucleotides that are just floating around, it's going to bring them to their complementary bases. So remember, A and T are always going to bind together, and C and G are always going to bind together. So if we have a DNA strand that goes A, T, C, T, G, we're going to bring in a T to match with the A, an A to match with the T, a G to match with the C, an A to match with this T, and a C to match with this G. And that's what this DNA polymerase is doing. It's bringing these free nucleotides that are just floating around, and it's matching them with their complementary bases. And we're going to have these two strands right here. So while this DNA helicase is unwinding our DNA, this DNA polymerase is going to be building a new strand of DNA. We're going to have a leading strand and a lagging strand. And you don't really need to understand everything that's going on here, but let's just say that each strand of DNA, so each side of this DNA, is going to have a direction. And this DNA polymerase is going to have to move in the direction of that DNA. So one strand is going to have a direction that makes the DNA polymerase move away from the helicase. The other strand is going to have a direction that makes this DNA polymerase move towards where that helicase is unwinding. So this leading strand is going to be moving towards the DNA helicase. It's moving in an unbroken strand right here. As the DNA is unwound, then we are adding these free nucleotides and building a new strand of DNA. This lagging strand, on the other hand, is moving away from where the helicase is unwinding. So this DNA polymerase has to start at a point on the unwound strand. It's moving away from where the DNA helicase is unwinding, and then it's going to come to a spot where it has already attached these complement base pairs. So then the DNA polymerase is going to have to jump down the DNA strand and start moving away from the helicase again until it gets to the point where it had started the last time. So we have this leading strand and this lagging strand. So we're building new DNA. One of the strands is from the original DNA, and then we are building a complementary strand onto that original strand of DNA. And then once we are done, we're going to have termination. This is the final stage in DNA replication. And this DNA replication is going to be complete when we have two strands that are identical, two identical strands of DNA. We're going to have one strand from the original DNA and then one new strand that was built by that DNA polymerase. That's going to leave us with two identical strands of DNA. We have finished our DNA replication.